All right, we're going to talk about determining the structure, the three-dimensional structure of DNA. So we're going to talk about a few scientists. Now, if I asked you the structure of DNA, you should be able to already tell me that it is a double helix. You should be able to draw it like that. The line represents the sugar phosphate backbone. The nitrogen bases are going to go towards the inside, and they are going to complementary base pair. Now, determining the structure of DNA involves some scientists. So these are your nitrogen bases. Nitrogen bases are going to complementary base pair. You have to remember A always is hydrogen binding with thymine, and guanine always hydrogen binds with cytosine. The next thing that we look at is how it is anti-parallel. The nucleotides are oriented 5' to 3'. This is anti-parallel. All right, the first scientist I want to talk about is a scientist by the name of Erwin Chargaff. Erwin Chargoff basically determined what we call the Chargoff rules. His job was to determine which nitrogen pairs are going to base. So in his experimentation as a scientist, he helped determine the 3D structure by learning that the amount of adenine is always equal to the amount of thymine. The next thing that Mr. Chargoff learned is the amount of guanine is equal to the amount of cytosine. So, A, hydrogen pairs with thymine, guanine, hydrogen pairs with cytosine. This is known as Chargoff's rules. All right. The next scientist that I want to discuss basically learned that the 3D structure of DNA is helical. So not necessarily the 3D structure, but basically that it's got this double helix shape. So we can say it's a double helix. It's got two sugar phosphate backbones, nitrogen bases face towards the inside, they hydrogen pair, and they are anti-parallel. So the scientist that found out this is accredited to two scientists. Rosalind Franklin and Maurice Wilkins. Now, what's special about Rosalind Franklin and Maurice Wilkins is they used X-ray diffraction studies. And what they discovered when they took X-rays, they said DNA is helical. Now, they didn't use the term double helix, they just said it was helical. So basically they took X-rays of DNA and noticed that it was helical. So the biggest and most important scientist that we're going to learn is going to coin the word double helix. And they're the ones accredited with finding out the actual 3D structure of the sugar phosphate backbones with the nitrogen bases facing towards the inside. And their names are James Watson and Francis Crick. This was going down in 1953. Now, I have to write notes on here. I'm trying to write big so you can see. All right, the first thing that they did was they never used any of their own experiments. What they did was they used the information from Erwin Chargoff and Rosalind Franklin, and they basically deduced or came up with the structure of DNA based on the knowledge that was known. They used evidence from others, mostly from Rosalind Franklin and Maurice Wilkins and Erwin Chargaff. So, this is what they said. Now, they did propose that this is a double helix model. They're the ones who actually said the DNA is a double helix, which I just drew. So, they used the information from Erwin Chargoff and Rosalind Franklin and Maurice Wilkins to come up with this double helix structure. Now, with this, they basically said there are 2 sugar phosphate backbones, which we've learned. The next thing, nitrogen bases. Point towards the inside of the model for the molecule. Nitrogen bases, complementary base pair. The 2 strands of nucleotides are anti-parallel. One strand goes from 5' to 3'. The other strand is oriented 3' to 5'. And because they said that Rosalind Franklin said that they're helical, they said the 2 strands wrap around each other to create a helical shape. So that's how we determined the 3D structure of DNA.